The spikes make the stickleback hard to eat. But the lake sticklebacks have lost those spikes on their bellies. And it's this that intrigues researchers David Kingsley and his colleague Dolph Schluter. To understand what's behind it, they first identified the gene that makes the stickleback's spikes. It's one of those key body plan genes, and not surprisingly, they found it to be identical in both the ocean and the lake stickleback. The question was, why hadn't it been turned on in the lake stickleback, which had lost its spikes? Kingsley felt the answer might lie in a switch.
We know these genetic switches exist, but they're still very hard to find. We don't have a genetic code that lets us read along the DNA sequence and say there's a switch to turn a gene on in a particular place.
But eventually, hunting through the vast stretch of DNA that does not code for proteins, he found it. A section of DNA that had mutated in the lake stickleback. These mutations meant that the switch was broken. It didn't turn on the gene that makes spikes. But this work may have implications far beyond sticklebacks. They are convinced that there is a link between the stickleback losing its spikes and other creatures, like a manatee losing their legs. And they have two tantalizing clues. One, the same body plan gene that is responsible for the stickleback spikes also plays a role in the development of the hind limbs. The second clue is more tentative. The lake stickleback may have lost its spikes, but evolution has left behind some tiny remnants. The traces of bones. And they are lopsided, bigger on the left than on the right.
We thought, wouldn't it be amazing if in fact this classic unevenness is the signature of using the same gene to control hind limb loss in incredibly different animals.
So Kingsley and his team went looking in manatees, searching for this lopsided pattern. And they found it. In box after box of manatee skeletons, they saw pelvic bones that were bigger on the left and smaller on the right. Right now, Kingsley and his team were looking for the same switch in the manatee that caused the lake stickleback to lose its spikes. And if they find it, they will have a powerful explanation for something that baffled Darwin. How creatures like manatees, whales, and snakes can evolve away their legs. But all this begs another question. If switches can play such a profound role in the different shapes and patterns of animal bodies, from wing spots to spikes to hind legs, what is throwing those switches in the first place? Researchers would see the answer in animals very familiar to Darwin, those Galapagos finches. Arkat Abzhanov and Cliff Tabin have spent years trying to find out exactly how those Galapagos finches got their different beaks. Their starting point was what they had learned from Darwin himself. Their beaks were vital to the bird's survival. On an island where the main food was seeds, finches had short, tough beaks for cracking them open. On an island where the main food was from flowers, birds had long, pointy beaks for sucking up nectar and pollen. And they knew something else. The finches are born with their beaks fully formed. So the answer to why they had such different beaks must lie in something that happened to them as embryos in the egg.
Something amazing is happening inside those eggs. Genes are turning on and turning off. And depending on exactly how they turn on and off will determine what type of finch is formed.
To find out just what was going on, the researchers first had to collect some eggs.
There she is. So the female just came back. Yeah. Just in the eggs. So it's very likely that she already has a clutch. Great. She's coming out.
Abzhanov checks a ground finch nest and finds a single egg. He won't remove it because the mother might abandon the nest. Another nest already has 3 eggs. He takes one for his research, as he knows the mother will lay a replacement. The team collects several eggs, with embryos at different stages of development. That way they will be able to chart exactly how the different beaks grow. Back in the lab, they can begin the process. This cactus finch embryo is well on the way to its signature long, pointy beak. And this ground finch embryo is growing a short, thick beak.
What we wanted to do was try and understand the genes that were involved in making the beak the way it was, making a big, broad, thick beak different from a long, thin beak or a short, thin beak.
They concentrated on a group of genes known to control the growth of birds' faces. As they looked, they saw something intriguing. One particular body plan gene became active in the ground finch with the short, thick beak on the 5th day of development. But it didn't go to work in the cactus finch with its long, slender beak for another 24 hours. This was a revelation. The same genes were responsible for the beaks in all types of finch. Any differences were in timing and intensity.
We've got it. We nailed it. It's the same genes in making a sharp, pointy beak or a big, broad, nut-cracking beak. What's essential, what makes the difference, and all the difference, is how much you turn a gene on, when you turn it on, when you turn it off.
And the revelations didn't end there. There was something special about this gene. Like all body plan genes, it doesn't actually make the stuff of our bodies. It didn't make the cartilage for the finch's beaks. It throws switches. And the switches then turn on or off the genes that do make the beak.
These are a different type of gene. They're genes that boss other genes around.
Scientists now realize that not all genes are created equal. Some make the stuff of our bodies, and switches are needed to turn many of these stuff genes on and off. The body plan genes are what throw these switches, which tell the stuff genes what to do and when. This subtle choreography can have profound effects on how different animal bodies are formed. And this knowledge is helping us solve perhaps the biggest Darwinian puzzle of all, the mystery of the great transformations. It all goes back to Darwin's idea of the tree of life, that all life forms are ultimately related, and from the earliest common ancestor over billions of years, they have changed and diversified, so that creatures that started out looking the same evolved to become completely different. And scientists have made some amazing connections. That dinosaurs share a common ancestor with birds, and that a fish must have been the ancestor of all 4-limbed creatures, even us. Of all his ideas, this was probably Darwin's most astonishing.
It was one thing to grasp how 2 species of finch could become different, how their beak shape could change. That was a small step. But what about the big differences? The differences, say, between the fish that swim in the sea and the animals that walk on land. How did those changes take place?
Over the years, evidence for these great transformations has been found. For instance, just a year after Darwin published On the Origin of Species, a fossil called Archaeopteryx was discovered. It had features of both birds and dinosaurs. And Darwin had seen equally persuasive evidence in embryos. Those slits in the ear of all land creatures, even humans. In us, they become tiny bones in the inner ear. But in fish, they become gills. A tantalizing hint that land animals must be descended from fish. But the stumbling block has always been how. How could a fish develop legs and walk on land? Darwin had no idea. But Neil Shubin was determined to tackle that problem.
It captured my imagination. I mean, here's a fin, and on the other side was a limb, and they looked different in many ways. And I thought, well, what a first-class scientific problem to devote my research to. And I've been devoting pretty much my research to it ever since, over 20 years.
The first stage in Shubin's quest was to find a fossil. If Darwin were right, somewhere out there had to be a transitional form. A fossil that was part fish, but had the beginning of legs. But where to look? He had one clue. The fossil record shows that creatures with legs first appeared some 365 million years ago. Before that, they were only fish. So summer after summer, Shubin set up camp on Ellesmere Island, just a few hundred miles from the North Pole. It has exposed rock from that crucial transitional time. The scientists' own video shows how remote and bleak the place was.
It's cold. It's about freezing every day over the summer. Winds are high. They can get up to 50 miles an hour. There are polar bears there. We have to prepare ourselves by carrying guns. It's a beautiful place. You gotta love it. It's my summer home.
Each expedition was costly, but after 3 of them, there was little to show for their efforts. A 4th trip seemed pointless.
I remember having a conversation with my colleagues, saying, well, should we go? Is this really a waste of money? This was our do-or-die moment, and we almost didn't go.
But they decided to try one last time. After 3 days, they still hadn't found anything. Then, just when no one was expecting anything to happen,
a colleague was cracking rocks, and I was working 5 feet from him. And I hear, hey guys, what's this?
Well, look what we found. Yes.
Sticking out of the cliff was the snout of a fish. And not just any fish, a fish with a flat head. And by seeing a flat-headed fish in rocks about 375 million years old, we knew we had found what we were looking for.
a flat snout with upward staring eyes, the signature of an animal that pushes its head out of the water. And for that, it would have needed something like arms.
What we did at that moment was all jump around, high-fiving. It was there were only 6 of us in the field that time, so it was quite a scene.
Back at home, Shubin and his team got to work, examining their 375-million-year-old fossil. They named their new finding Tiktaalik, an Inuit word for a freshwater fish. Tiktaalik is a perfect transitional form. Much of its body is that of a fish. It's covered in scales. But it also had something very unfish-like. An arm-like fin, or perhaps a fin-like arm. Tiktaalik had the bone structure that is seen in the arms and legs of every four-limbed animal. One big bone at the top, two bones underneath, leading to a cluster of bones in the wrist and ankle. It's the same pattern that is found in everything from sheep to sheepdogs, to Shubin himself.
You now have an animal that can push itself up off the substrate, either on the water bottom or on land.
One obvious question was, why had Tiktaalik evolved this new structure? One possible answer is suggested by other fossils found near it.
There are large predatory fish about 10 to 15 feet long living alongside Tiktaalik.
Tiktaalik was prey. To survive, it had few choices.
You can get big, you can get armor, or you can get out of the way.
Shubin thinks Tiktaalik got out of the way. With those arm-like fins, it could have dragged itself to safety on land or in the shallows. But this was only half the answer.
What it doesn't show us is the actual genetic mechanism, the genetic recipe that builds a fin into that which builds a limb.
At 375 million years old, Tiktaalik's DNA had vanished long ago. Shubin needed a next of kin, a fish relative that was still alive.
What we needed was a creature that was in the right part of the evolutionary tree, but also a fish that has a very fleshy fin. So the search was on.
A number of fish fit the bill, but Shubin favored one in particular: the paddlefish.
Paddlefish is a really weird fish. They develop this really long snout, and they're really voracious. They eat each other. So oftentimes you'll lose a lot of your fish when they swim together because they'll eat each other.
Living in the shallow waters of the Mississippi, it's also a living fossil. Scientists have spent years working out the relationships between different species of fish, and they know that the paddlefish is one of the last survivors of the class to which Tiktaalik once belonged. But unlike Tiktaalik, the paddlefish is in plentiful supply.
Paddlefish is a common source for caviar, so we'd get our paddlefish from caviar farms.
Intriguingly, even though Tiktaalik is extinct, the paddlefish is actually the more primitive form. Its fins bear far less relation to an arm or leg than Tiktaalik's. And because they are related, the two kinds of fish should share the same genes. So Shubin began looking at paddlefish embryos, hunting for the genes that built its fins. And soon, he zeroed in on one particular group of body plan genes, called Hox genes. Hox genes have been found in all complex animals, from the velvet worm that dates back some 600 million years to the modern human. And in all that time, the letters of their DNA have remained virtually unchanged. They are aristocrats of the gene community, near the very top of the chain of command. They give orders that cascade through a developing embryo, activating entire networks of switches and genes that make the parts of the body. They are absolutely critical to the shape and form of a developing creature.
These genes determine where the front and the back of the animal is going to be, the top, the bottom, the left, the right, the inside, the outside, where the eyes are going to be, where the legs are going to be, where the gut's going to be, how many fingers they're going to have.
Shubin found that Hox genes had a key role in the formation of paddlefish fins. One set of Hox genes orders the first stage of fin development, a sturdy piece of cartilage that grows out from the torso. Amazingly, in all four-limbed animals, even us, exactly the same genes create the long upper arm bone. In the paddlefish, another set of Hox genes command the next stage of fin development. Again, exactly the same genes control the growth of our two forearm bones. Finally, the same genes, working in a different order, make the array of bones at the end of the fin. The same sequence of the same genes makes our fingers and toes. This was a massive revelation. Suddenly, the origin of creatures with arms and legs didn't seem such a huge leap after all. If the same genes were at work in Tiktaalik, then many of the genes needed to make legs and arms were already being carried around by prehistoric fish. All it needed was a few mutations, a few changes to the timing and order of what was turned off and on, and a fin could become a limb.
Oftentimes, the origin of whole new structures in evolution doesn't involve the origin of new genes or whole new genetic recipes. Old genes, old genetic pathways can be reconfigured to make marvelously wonderful new things.
So it is now possible to answer what Darwin didn't know and explain how all four-legged creatures could be descended from fish. Around 375 million years ago, a creature like Tiktaalik was under attack, harried by predators. But some random changes to the activity of the Hox genes led to its fins, developing a structure like a limb. Tiktaalik could now haul itself out of danger onto dry land. On land, it would have found a world of plants and insects, a world ripe for colonization, a world perfect for animals with arms and legs. And so, over millions of years, these new limbs evolved, changed, and diversified. Some became adapted for running, others for flying, some for digging, others for swinging. And so four-limbed creatures took over the world in a multitude of different ways. And all because of some changes to an ancient set of genes. And this is the true wonder of where our new understanding of DNA has led us to. There are genes that make the stuff of our bodies, switches that turn them off and on, and still other genes that give those switches orders. Together, in a complex cascade of timing and intensity, they combine to produce the amazing diversity of life on this planet. That, truly, is something that Darwin never knew. But can this new science also explain perhaps the most fundamental question of all? What makes us human? The scope of human activity is simply astounding.
What fascinated me were all the crazy things that humans do. You look around the world, and if there's something bizarre and interesting that you could be doing, humans are up to it somewhere in the world. And when you look at all of this, you just have to ask yourself, what makes us special? What is the basis for this humanness?
For all nature's wonders, the achievements of the human mind are truly unique. We are the only species to think about what others think about us, to punish those who have harmed others, to create art, music, architecture, to engage in science, medicine, the microchip, only we can destroy millions at the push of a button. Hardly surprising, then, that for centuries we thought that humans were different from all other species, better, created in the image of God. But then Darwin began to draw conclusions from evidence like gill slits in human embryos that showed that we were descended from fish. But it was when he drew parallels with other close relatives that he got into real trouble.
Shortly after Darwin returned from his voyage, in London, an orangutan named Jenny went on exhibit. And this was a huge sensation. This was the first great ape to be exhibited in captivity. And Darwin was absolutely taken with how she was sort of childlike in her ways. And he saw a lot of human behavior in the way this orangutan behaved.
When Darwin suggested that human beings must actually be descended from apes, he was savaged. He was accused of attacking that core belief that humankind had been created in the image of God above all other creatures. But today, the idea that we share a common ancestor with apes is completely accepted in biology. Instead, as a result of having sequenced the genomes of both humans and apes, we face a very different puzzle. Katie Pollard is an expert on chimp DNA.
Given all the obvious differences between humans and chimps, you might expect our DNA to be really different. But in fact, it's more like 99% identical.
Just a 1% difference in the DNA of humans and chimps. The mystery facing modern science is not how can such different animals be related, but how can such closely related species be so different? That really is something that Darwin never knew. But slowly, scientists are starting to find the answers. And one answer begins with insights into the genetics of a key human organ, our hands. The human hand is a marvel. Nimble and dexterous, nothing quite like it exists anywhere else in nature. It offers us a unique combination of precision and power. And much of that is down to one particular digit, our thumb.
One of the features of the human hand is our ability to touch all 4 fingers with the thumb. And that allows us to make grips like this. Gives us a lot of precision. The power grip is the ability to put a lot of strength into this sort of contact. So if you're holding a ball, you're basically pinching it. And we can put a lot of strength into that.
The better to throw a fastball with. Finding out why we have such versatile hands compared to our nearest relatives is the task of Jim Noonan at Yale University. He began sifting through that vital 1% of DNA that is different in humans from chimps.
It's kind of one of the fundamental questions in science is what makes us who we are. That's really what we're trying to get to. What makes humans human?
It was slow work. 1% may not sound like much, but it's still some 30 million of DNA's chemical letters. A's, T's, C's, and G's.
The genome's a big place. And just by looking at sequence, you really can't tell, for the most part, what is important and what isn't.
But eventually, in human DNA, he spotted something. A sequence that was different in 13 places compared to chimp DNA. The trouble was, he had no idea what this piece of DNA actually did. To find out, he inserted it into the embryo of a mouse. To make the effects of the DNA easier to follow, he attached it to another gene that gives off a blue color. That way he could see where the gene became active in the embryo. As the embryo developed, the piece of DNA seemed to be active all over the place. But most intriguingly, it was doing something in the growing paw.
Well, I thought, wow, this is really cool. It was a really, really striking image.
What Noonan saw was that the human DNA became active in the mouse embryo's thumb and big toe. It seems that Noonan may have found a switch that helps form that key human attribute, our thumb. The part of our hand that gives us so much power and precision. It's that power and precision that enables us to hold a paintbrush, manipulate tools, pilot a jet fighter, record our thoughts. All those things that separate us from other apes. Of course, having a nimble hand is one thing, but you have to know how to use it. And for that, you need to have humankind's other signature organ, our brain. The human brain is vast, 3 times bigger than a chimp's, and is structured very differently. How this extraordinary organ evolved is central to understanding why we are the way we are. It is something that Darwin himself was at a loss to explain, which is why many of his critics remained unconvinced by his account of human origins. But now, part of the answer to why we have such a remarkable brain may have come from a surprising source. Hansel Stedman is a dedicated athlete and a medical doctor. He never imagined he would come up with an answer to a profound evolutionary mystery. He has devoted his career to trying to cure muscular dystrophy, a distressing and sometimes fatal degenerative disease. His quest is very personal.
My first exposure to muscular dystrophy was inescapable. My younger and my older brother, both born with muscular dystrophy.
Muscular dystrophy is a genetic disease. Its sufferers have a mutation in one gene that robs their muscles of the ability to repair themselves.
Typical workout here on the rocks might blow through a few thousand muscle cells, but they'll regenerate overnight, and if anything, be a little stronger the next day I come in as a result of all of that. Whereas in muscular dystrophy, the injury process is greatly accelerated, and the injury process outstrips the body's ability to repair.
In search of a cure, Stedman is investigating the hundreds of genes that control the development of muscles. So when the Human Genome Project took off, Stedman seized his chance.
When the horsepower of the entire Human Genome Project kicked in, we knew exactly what to look for.
Stedman was hunting for any new muscle-making genes. And so, as the human genome was sequenced, he began sifting through the vast mountains of data. Eventually, he found what he was looking for. A previously unidentified muscle-making gene. But there was something strange about this new gene. It didn't look like any other muscle-making genes. 2 letters were missing. This gene should cause a disease.
It became very clear early on that if you have a mutation of this type, you get some serious muscle problem going on.
Here was a puzzle. Why would humans carry a gene that was clearly damaged? Perhaps it was simply a mistake in the data. Stedman decided to dig a little deeper and look in another human subject.
In the Department of True Confessions, we do certain experiments first on ourselves, largely out of convenience. You know, you can swab your own cheek and get working on some DNA.
To his utter amazement, he found the same damaged gene in himself.
I'm seeing this in my own DNA, and it's suggesting, no, wait a minute, that means there's a muscle disease here somewhere, a muscle disease that I'm unaware of, and I thought it would be worth checking this out in some other members of the lab.
A few swabs later, and
sure enough, at the end of the day, every single person had the same glitch and their same DNA at the same place.
Here, then, was a real mystery. It seemed that this particular muscle-making gene was common in humans. But when he identified the same gene in apes, it was just like any other muscle-making gene. Why was there such a difference? What did this gene enable one species to do that the other could not? Stedman began to research the role of this gene in apes, and he found it made one particular kind of muscle. The muscle for chewing. In fact, the muscle used to close the jaw. In humans, that genetic glitch meant that we chew with just a fraction of the force of an ape. This in itself was interesting, but where Stedman went next was truly intriguing and highly controversial. He drew a direct connection between the power of our jaw muscle and the evolution of the human brain. Stedman's thinking goes like this. The skulls of apes and humans are made of several independent bone plates. They let our heads get bigger as we grow. The muscles for chewing pull against these plates. And in an ape, these forces can be enormous.
In the gorilla, the muscle, the size of a human thigh muscle, lives here and has to go through this large space to power the jaw moving back and forth. We're not talking biceps, triceps, we're talking quad here. This is an enormous muscle that has to come right through this hole here to power the jaw closing apparatus.
Stedman contends that all this muscle power forces an ape's skull plates to fuse together at an early stage. And this puts limits on how much the brain can grow.
In the chimpanzee, gorilla, orangutan, those growth plates are pretty much shut down, closed for business by about 3-4 years of age. In a human, they remain open for growth to perhaps age 30.
This, Stedman believes, is the key. A mutation in our jaw muscle allows the human skull to keep expanding into adulthood, creating a bigger space for our brain. And so our most important organ is able to grow.
It's very cool to us to think that some kind of muscle-altering mutation might have actually been a signature event in the evolution of what makes us distinct as a species. It might have been an absolute prerequisite for us landing where we are today.
But having the space for a big brain is one thing. What is needed to actually grow one?
And again, a very small cerebral cortex with very few.
That is the question that Chris Walsh is trying to answer. He's another scientist who never expected to be taking on what even Darwin didn't know.
I never thought that I'd be studying evolution. I'm a neurologist interested in the brain and in kids with neurological problems.
How you doing, buddy? You doing all right? Huh? Doing okay?
No one was more surprised than us to find that the study of kids with disabilities would lead us into these fascinating evolutionary questions.
Is breathing generally okay during the day?
Sometimes when he gets startled, it'll go up fast, like, but then he calms himself right back down and it comes back.
Walsh is a specialist in a rare disorder called microcephaly. Children with microcephaly are born with brains that can be half the normal size.
This disorder can be very devastating for the kids that have it. They typically will have severe mental retardation and so will not be able to achieve normal language and normal schooling. And so it's really an event that defines the whole family. It defines the lives not only of the child, but of the parents of that child. And these families are desperately eager to try to understand at least what caused the disorder in their kids.
The purpose of Walsh's work was initially to help families that might be carrying any defective genes causing microcephaly to plan their lives.
We're able to offer those families predictive testing so that if they're planning on having additional children, we can tell them ahead of time whether that child is likely to be affected or not.
First, Walsh had to decide where to look in the vast genome to find any possible microcephaly-causing genes. So he focused on one particular area of DNA, Other research suggested it contained a gene involved in the condition. That gene is known to control how and when brain cells divide in animals, such as fruit flies and mice.
What this gene seems to do is help control the fundamental decision that the brain has to make, which is, when do I stop making cells? When is the brain big enough?
Then his team began searching for that same gene in a family with a history of the disease. And sure enough, they found something. A gene that helps direct brain growth. And crucially, it was defective. Walsh decided to check this finding in other patients.
Once we found this gene, we sequenced it in our kids with the microcephaly disorder, and we found that one family after another had a disabling change in the gene that completely removed its function.
In total, he has found some 21 different mutations responsible for microcephaly. Sometimes one of the DNA's chemical letters is replaced with another letter. Sometimes letters are missing entirely. But whatever the defect is, they all stop the brain cells from dividing at a very early stage of development. Walsh was now certain, thanks to his microcephaly patients, he had found a gene key to the growth of the human brain. Now he decided to compare normal versions of the gene found in healthy humans with the same gene in chimpanzees, our closest relatives. And what he found was astonishing. The gene in humans was radically different from that found in chimps. There had been a large series of mutations. It could be that these mutations were a major factor in the evolution of our huge brains. And this discovery came about only because of Walsh's work with his patients.
I think one of the amazing things for us was the extent to which studying human disease can unexpectedly enlighten us about something like human evolution.
But this is only the beginning of our understanding of the evolution of the human brain. It's an area of research that is now attracting scientists with a range of skills that Darwin would have marveled at. Katie Pollard is a biostatistician. Her life is spent crunching numbers.
What I love about my work is geeking out on a computer and writing programs and thinking about biology. And that in doing this, I'm actually working on something that not just scientists care about, but really every human being can relate to and cares profoundly about. And that's what makes us human.
Pollard has constructed an ambitious computer program. It's designed to highlight DNA that is similar in apes and other animals, but which is very different in humans. That way, she hopes to identify the key DNA that makes us, us.
Out of these 15 million letters that make humans different from chimps, we need to try to figure out which ones were important. And so we use a technique which is to look for places where human is different from chimp, but chimp looks almost identical to the other animals.
She too is looking for DNA relating to the human brain.
The brain is one of the things that's changed the most during human evolution, both in terms of its complexity and its size. And so when we look to find the parts of our genome that make us human, we're particularly interested in finding out whether these are things that are involved in the brain.
It is a huge feat of number crunching. As Pollard loaded in DNA sequences from both humans and chimps.
You basically take a bunch of computer hard drives and you stack them up together. We were able to take a task that would have run for 35 years on a desktop computer and do it in one afternoon.
And at the end of that afternoon, they had a whole array of material charting the differences between humans and chimps. Importantly, many of those differences were not in the actual genes. They were in switches.
It turns out that the vast majority are not genes. Instead, they're pieces of our DNA that we can think of as switches. They're pieces of DNA that turn a nearby gene on or off, that tell it where, in what cells in our body, in what tissue, at what time, or at what level to be operating.
And there was something even more intriguing about those switches.
A large number of them, more than half, were nearby a gene that was involved in the brain.
In Pollard's work, one particular piece of DNA stood out. It was a piece of DNA that is known to be active in the development of one of the key parts of the human brain in the embryo, the cortex. The cortex is that wrinkled outer layer of our brain. It's vital for those defining human capabilities like language, music, and mathematics. When she looked at that DNA in chimps and compared it to the same DNA in a chicken, it was different in just 2 letters. But in humans, it was different by 18 letters. A massive mutation.
This was about as great of a eureka moment as you could have as a scientist.
So here is another intriguing piece of evidence suggesting how DNA can shape our distinctive human qualities. We now know that DNA works in many different ways. Through genes that make the stuff of our bodies, through switches that turn those genes on and off, and through sequences of DNA's chemicals that throw those switches. Taken together, what this all adds up to is a way that we can at last understand how small differences in DNA can generate enormous change.
Basically, you can make massive changes just changing those switches. So a small change, a couple of DNA letters, could have a profound effect.
And so that final Darwinian puzzle, how a human can be so closely related to an ape and yet be so different, is now slowly being answered. "150 years after Darwin first put forward his grand theory to explain the great diversity of life, the scientists who carry on his legacy have advanced his work in wondrous ways."
I think if Darwin were here today, he'd be absolutely stunned, delighted, even moved to see how much his theory has grown.
What we now are able to understand, on the one hand, would just blow him away. But I also think it would give him enormous satisfaction because ultimately, everything we've been learning validates the things that he said.
I think that Darwin was a remarkable scientist and absolutely should be celebrated. However, I do not think that he was the end of evolution. On the contrary, I think he was the beginning. He outlined the major points, but we have discovered more than I think he would have imagined possible.
As we celebrate the 200th birthday of Charles Darwin and the 150th anniversary of his great work, there is still much more to understand about how the endless forms of nature have arisen. And in rising to that challenge, it is likely that we will continue to advance medicine and come to a better understanding of ourselves as well.
NOVA has a brand new evolution website with dozens of videos, interviews, slideshows, and the latest in evolutionary science. Bookmark it today and let us know what you think. Find it at pbs.org. Major funding for NOVA is provided by the following. ExxonMobil, taking on the world's toughest energy challenges. And by David H. Koch. And
discovering new knowledge. HHMI.
And by the Corporation for Public Broadcasting, and by contributions to your PBS station from viewers like you. Thank you.
Additional funding by Promega Corporation. Discovering every day. Promega, the science of life.
And by the Wisconsin Alumni Research Foundation in support of the University of Wisconsin.